Okay, I've split this into two videos. So we're just reviewing what we just talked about in transcription by looking at this diagram on page 429. Let's see, this is figure 15. So transcription, remember trans, like across something, and script meaning to write. So it's the act of writing something down, writing that genetic code down to move it across the nuclear membrane to get it out of the nucleus and into the cytoplasm. All right, so transcription is the first step of making a protein. During transcription, the sequence of nitrogen bases on a gene determines the sequence of bases on messenger RNA. So let's see what happens here. So here we have the DNA, okay, and here's the other half of the DNA. It's opening up, and as it opens up, a protein, and we're not going to talk about this again, you'll learn more about this in biology, but a protein comes along here and it attaches. So the messenger RNA nucleotides, see, this is messenger RNA, okay, the nucleotides pair up with the DNA nucleotides. So it's so there's a protein here that's taking these RNA nucleotides and attaching them, making this, and they're not showing this protein, but anyway, it's making this strip that's like a half of the helix, right? Because the RNA doesn't have the other half on it. It's just one part of it. All right, so this completed messenger RNA is now going to move out into the cytoplasm through the nuclear membrane into the cytoplasm, and let's see what happens from there. Okay, so here's the messenger RNA that just came out of the cytoplasm, and it's going to get snatched up by one of these ribosomes, and the ribosome is going to use it to create a protein. And so how does that happen? Well, the transfer RNA, which is this looking thing right here, this kind of pink thing right here, this is called transfer RNA. This transfer RNA carries the amino acids, and you can see these listed here. This is like A, U, and G. So these are paired up. These are these are the code, the kind of the opposite of the codes, right? So if we've got a code on our messenger RNA that's UAC, the code that's on the transfer RNA is the opposite, is AUG. These are still the nucleotides. But this transfer RNA is attached to this amino acid. Okay, so this particular amino acid has a transfer RNA with the code AUG, and it's going to attach itself to the messenger RNA. It's going to be basically pulled in at UAC, and it's a brief attachment while the ribosome's connecting these amino acids. So this is what happens. The transfer RNA comes in with its code out towards the messenger RNA. It gets connected. They don't want this to be attached if it's got the wrong code. So we've got to connect it, and then while that's briefly connected, the ribosome will attach these amino acids together. And then after these are attached, the transfer RNA just goes bye-bye, and it goes and does it again. All right, so let's take a look at that. So the ribosomal RNA, the rRNA, helps form chemical bonds that attach one amino acid to the next. So these are being attached with the help of the ribosome. The first transfer RNA separates here from its amino acid, so it's going bye-bye, and from the messenger RNA. So see, when I said it was briefly attached to these codes, they were briefly attached to each other just long enough to get those amino acids to bond. Okay, so then a third transfer RNA is coming in. So there's one bonded each time, or two bonded each time. So there's leaving, another one, as this one's leaving, another one's coming in. They're going to bond these two amino acids together. All right, and as they do this, this long chain of amino acids is created, and it's going to create a protein. So let's talk about that. All right, again, this box up here, that's representing our amino acids. This is our transfer RNA molecule, and this is our nucleotides, 3 different nucleotides. And remember, those are always representing adenine, guanine, cytosine, or uracil, not the not the thymine, but the uracil instead of thymine. Okay, so 3 types of RNA. On the previous page, you read about messenger RNA, and there are 2 other types of RNA, transfer RNA, tRNA, and ribosomal RNA, which is rRNA. So figure 16 that we just looked at illustrates how the 3 work together to make proteins. The process of making a protein from RNA is called translation. Translation occurs in ribosomes. Recall that ribosomes are cell organelles that are attached to the rough endoplasmic reticulum, and ribosomes are also in a cell's cytoplasm. So they can be free-floating or they can be attached to the endoplasmic reticulum, and particularly the ones that they attach to are called rough. Okay, so rough just means that they have ribosomes on them. There are also smooth endoplasmic reticulum that do not have ribosomes attached. All right, so translating the messenger RNA code. Making a protein from messenger RNA is like using a secret code. Proteins are made of amino acids. The order, this is big, the order of the nitrogen bases in the messenger RNA determines the order of the amino acids in a protein. 3 nitrogen bases on a messenger RNA form the code for 1 amino acid. Each series of 3 nitrogen bases on messenger RNA is called a codon. There are 64 codons, but only 20 amino acids. So some of the codons code for the same amino acid. One of the codons codes for an amino acid that is the beginning of a protein. This codon signals that the translation should start. 3 of the codons do not code for any amino acid. Instead, they code for the end of the protein. They signal that translation should stop. Reading check. What is a codon? So a codon is, a codon is a series of 3 nitrogen bases on the messenger RNA. The part that is on the transfer RNA is called an anticodon. Okay, it's the opposite of the codon. It's the pairs that match the codon. Mutations. You have read that the sequence of nitrogen bases in DNA determines the sequence of nitrogen bases in messenger RNA, and that the messenger RNA sequence determines the sequence of amino acids in a protein. You might think these sequences always stay the same, but they can change. A change in the nucleotide sequence of a gene is called a mutation. The 46 human chromosomes contain between 20,000 and 25,000 genes that are copied during DNA replication. Sometimes mistakes can happen during replication. Most mistakes are corrected before replication is completed. A mistake that is not corrected can result in a mutation. Mutations can be triggered by exposure to X-rays, ultraviolet light, radioactive materials, and some kinds of chemicals. Here are the types of mutations. There are several types of DNA mutations. 3 types are shown in figure 17. In a deletion, think about what the word delete means, to take away something. In a deletion mutation, 1 or more nitrogen bases are left out of the DNA sequence. In an insertion, think about what the word insert means, to put something in. In an insertion mutation, 1 or more nitrogen bases are added to the DNA. In a substitution mutation, think about what substitution means. Put something in while you take something else out. 1 nitrogen base is replaced by a different nitrogen base. So each type of mutation changes the sequence of nitrogen base pairs. This can cause a mutated gene to code for a different protein than a normal gene. Some mutated genes do not code for any protein. We talked about that, the junk DNA, right? For example, a cell might lose the ability to make one of the proteins it needs. Okay, so some mutated genes do not code for any protein, but let's look at this. So mutations, here's an example of the 3. So here's our original, and here something has been substituted. So instead of reading CGA, the original read, now it reads CAA. So the G has been replaced or substituted with an A. Okay, and the C that was connected to the G on the other side of the DNA strand is now a T because it's connected with an A. All right, so that's a substitution. An insertion mutation looks like. Okay, so an insertion is when these base pairs have been added. So here's your CGA that was there originally, and now we have CGTAGA. So we added an extra TAG there. All right, and then for a deletion, pairs have just been taken away. So the 3 base pairs have been removed. Other base pairs will move in to take their place. Okay. So what are the results of these? The results of a mutation. The effects of mutation depend on where in the DNA sequence the mutation happens and the type of mutation. Proteins express traits. Because mutations can change proteins, they can cause traits to change. Some mutations in human DNA cause genetic disorders, such as those described in Table 4. However, not all mutations have negative effects. Some mutations don't cause changes in proteins, so they don't affect traits. Other mutations might cause a trait to change in a way that benefits the organism. Key concept check. How do changes in the sequence of DNA affect traits? Well, changes in the sequence of DNA can have negative effects, beneficial effects, or no real effect at all on the organism's traits. So scientists still have much to learn about genes and how they determine an organism's traits. Scientists are researching and experimenting to identify all genes that cause specific traits. With this knowledge, we might be one step closer to finding cures and treatments for genetic diseases. Okay, the first example that they give is a defective gene on chromosome 12, the PAH gene. And this is something that calls phenylketonuria. So people with a defective PAH genes cannot break down the amino acid phenylalanine. If phenylalanine builds up in the blood, it poisons nerve cells. This is something they check babies for in the hospital. Every time a baby is born in the hospital, they will take a tiny little bit of blood from their heel to check for this, to make sure that they don't have this disorder. Looking at the second one, there can be a problem with chromosome 7, the CFTR gene. This causes cystic fibrosis. So in people with defective CFTR genes, salt cannot move in and out of cells normally. So mucus builds up outside of cells, and the mucus can block airways in lungs and affect digestion. Chromosome 7, the elastin gene, can cause Williams syndrome. People with Williams syndrome are missing part of chromosome 7, so it's a deletion, including the elastin gene. And the protein made from the elastin gene makes blood vessels strong and stretchy. And then chromosome 17 has a BRCA1 gene, and chromosome 13 has a BRCA2 gene. And these can cause types of cancers in women, breast cancer and ovarian cancer. So defect in the BRCA1 and the BRCA2 does not mean that the person will have breast cancer or ovarian cancer, but people with defective BRCA1 and 2 genes have an increased risk of developing breast cancer and ovarian cancer. On page 433, number 1 in the lesson review says, distinguish between transcription and translation. If you look at that root word script, remember that's like writing, like the messenger RNA is being written inside of the nucleus. And translation, it's kind of like when someone tells you, like if you're a Spanish and an English speaker, and someone tells you what the Spanish is and you translate it, you put it in another form. Okay, so remember, translation is taking in the messenger RNA by the ribosomes and creating another form going out, which is the amino acids. So transcription is the process of making messenger RNA from DNA, and translation is the process of making a protein from messenger RNA. Number 2, use the terms DNA and nucleotide in a sentence. Nucleotides make up the teeth, or you could think of them as the rungs of the ladder of the DNA. So the teeth of the zipper or the rungs of the ladder, that's what the nucleotides do. They make that up. They're the parts of the deoxyribonucleic acid, the part of that that's in the middle there. The nucleotides, actually the nucleotides are making up all of it. I'm kind of unsure why they put that in their book. They make up all of it in the sense that the DNA is they're connected nucleotides together. The part that's in the middle are the bases of the nucleotide. The rungs and the teeth of the zipper are the bases of the nucleotide. So, okay, well, let's go on to number 3. I can disagree with the book. A change in the sequence of nitrogen bases is a gene called a what? It's called a mutation, right? So a change in the sequence of nitrogen bases is just in the gene is called a mutation. Understanding key concepts. Number 4. Where does the process of transcription occur? Remember, you write down the recipe at your grandma's house. So in the nucleus is the answer, in the cell nucleus, C. Number 5, make a drawing that illustrates the process of translation. And if I was going to do that, I would probably show something like a ribosome with maybe the messenger RNA in here. And I could show different colors representing the different bases sticking out of the messenger RNA. Let's put 3 of them in here. So something like that. And then I might have transfer RNA that's connecting. Let's see. Let's do yellow and purple together. And red and green. And so maybe this transfer RNA connects all of these. And at the top, the transfer RNA has an amino acid, right? Some kind of amino acid that it's connected to. And so during translation, this ribosome, which is has 2 parts here, is reading the messenger RNA and taking in transfer RNAs that are attached to amino acids. And so the amino acids are growing in a long line of amino acids here. And so these amino acids come together to make a chain of amino acids, and that's originally going to become a protein. Oops, Protein. All right, so there's my okay. Number 6 says, distinguish between the sides of the DNA double helix and the teeth of the DNA double helix. Well, the sides of the DNA double helix are made of the sugar phosphate groups, and the teeth of the DNA double helix are made of the nitrogen bases. Number 7 says, identify the products of what process are shown in the figure below. Let's take a little closer look at the process shown in the figure. It looks like the DNA is coming apart and either side is getting its partner replaced. So it looks like this is a picture of replication. This is a that's making new DNA. Remember, it keeps this part of the DNA and is adding the other side. This part is being kept from this side and it's adding this side to it. Okay? So half of each new strand of DNA is made up of the old strand. Number 8. sequence. Draw a graphic organizer like the one below about important steps in making a protein, beginning with the DNA and ending with the protein. So first you're going to say DNA, from the DNA, and through transcription, messenger RNA is made. And then the transfer RNA is going to carry the amino acids to the ribosome on the messenger RNA. And then the ribosomal RNA is going to bond those amino acids together, and the amino acids in a long chain will eventually become the protein. All right. Number 9, hypothesize what would happen if a cell were unable to make messenger RNA. Well, if it wasn't able to make messenger RNA, no proteins could be made and the cell would die. Number 10, what is the importance of DNA replication occurring without any mistakes? Why is it important for it to occur? Well, the error-free DNA replication ensures that each new cell carries the identical genetic information as the cell that it came from. Remember that the DNA replication takes place right before mitosis, so that the new cells being made will be just like the old cells. It also occurs right before meiosis, when it's going to divide up that DNA and put it into to gametes, into egg and sperm. And so it's important that the right messages are passed on.